you. Good morning, everyone. My name is Minjin Park, and I'm a PhD student from Seoul National University. It's my honor to give a presentation, especially the last presentation of this session. Today I want to talk about the whole genome sequencing of Greenland muskox individuals using surface deposited skeletal elements. So let's imagine that you arrive in the Arctic tundra ecosystem. You may see some bare ground lakes or glaciers, and you may encounter this kind of species, the muskox. So first, I would like to introduce what a muskox is. The muskox is large herbivorous even-toed ungulate species living in the Arctic. Adult muskox, and they normally grow up to 300 kilograms for male and 200 kilograms for females. They graze on Arctic willow and graminoids, and they are preyed upon by Arctic wolves, polar bears, and brown bears. On YouTube, you may see the wonderful videos attacking wolves and defending muskox and themselves. Another interesting characteristics are their appearance. They have double coat and the outer layer of thick, and the long one is guard hair, and the guard hairs protect from the insects or shed water away from the skin. And beneath, the under soft down wool is cavit, and the cavit is naturally shed away from the skin in the spring, and some farmers can comb the cavit off the animal, and then they can make into yarns. And ever-growing longhorns is another wonder. The headgear for the male is for the male rotting display. The distinctive scent is why they are called muskox. Is secreted from the male reproductive gland. It is used for attracting females or marking territories. Then I want to introduce where they live. The current distribution is that they are endemic in Canadian mainland, Canadian Arctic archipelago, and north and east Greenland. But they are reintroduced in Alaska, West Greenland, Scandinavia, Quebec, and Siberia within 100 years for various purposes. These numbers are approximate census size in each region, and these lines are the route of reintroduction. So I could say the main wild population is in North America and Greenland. So thanks to the country's wonderful work, the ecological modeling could infer where they lived in the long-term time context. 21,000 years ago, they lived all across the Siberia, but after last glacier maximum, the population in Eurasia really shrunk, and they migrated into North America, and now they exist only in North America. So this kind of process explicit model was validated by fossil records. You may see here the population in Europe, Asia, Beringia was collapsed one after another. You can see here the red lines, the yellow lines here. This means is the last fossil records in Asia. It was in northeast Siberia. That means they exist until 3,000 years ago. But at the same time, the abundance in North America was increased. So the population met different fates. So what we could see in the modeling was the fluctuations in abundance and the population distribution. And I believe population genomics would be highly helpful to infer the evolutionary history. Especially the first ancient whole genome paper was published recently, and I think it delivers the importance of well-preserved samples, because the ancient sample was dated back to 21,000 years ago, and it was discovered in Wrangel Island. It found that it was the outgroup of all existing populations, and it has higher diversity than now existing populations. So I want to deliver these two main questions. Which materials could be used for ancient population genomic studies? And the second one is, what could be inferred from the whole genome sequence of Greenland muskox? So first, I would like to thank the sample contributors. Korea Polar Research Institute operated the Greenland excavation from 2016, and it was relatively easy to find this kind of surface deposited skeletons on the ground. So we collected 5 samples from Cirrus Passet and 12 from Mid-Somerset and 9 from L Island. Cirrus Passet and Mid-Somerset is located in north part of Greenland, and L Island is located in the east part. The sample size is 5, 12, and 9, and they were carbon dated, and we found that it is quite young and within 300 pp. I don't want to go all the details, but I want to highlight the sample preparation step. From Greenland excavation, we collected bone samples, and we could drill them into powder, and we make DNA extraction and library step in Max Planck Institute and Canada University. We sequenced based on Illumina platform, and together with the published sequence data, we could make genotype panels, and we could look into population structure, gene flow, and demographic history. The first result is that we tested which skeletal part was the most efficient for sequencing. We tested horn, long bone, and teeth. This plot is showing which skeletons are best in DNA damage. It means after death, DNA damage would not be fixed. So C2T frequency is the typical attribute of ancient DNA or damaged DNA. And the libraries from teeth showed the highest damage, and the long bone next, and horn showed relatively intact. And the proportion of mapped reads to total reads was also the highest in horn libraries, and it means it really conserved in the endogenous DNA. Surprisingly, not over 95% was preserved in horns, but long bone and teeth was relatively low. This is somewhat amazing because the proportion was also really nice, and also the horn was not really selected for the DNA extraction because of the keratin. So it is quite amazing. And the second result is that the population structure inferred by principal component analysis. Thanks to the well-preserved samples, especially from horns, we could sequence 5 samples from Cirrus Passet, 10 from Mid-Somerset, and 9 from Ella Island. Together with the published data, we could detect 3 main clusters, 3 main populations: Canadian mainland, Canadian Arctic, and Greenland. Our 3 study sites were aligned all in Greenland cluster, and the explained variance of first PC was used to detect the two subspecies. They have remarkable genetic differentiation between the two subspecies, and the name is white-faced and barren-ground. And we could find more stratification within populations: Canadian mainland west and east, Canadian Arctic south and north, and Greenland north and east. And among them, North Greenland has some affinity with Canadian Arctic, especially the North Canadian Arctic. So we could say from the Canadian Arctic population, the Greenland populations were believed to be separated from Canadian Arctic, and then they dispersed independently. So we could say that there was a robust genetic barriers between Canadian Arctic and Greenland. Even though the population abundance in North America increased over time, but there was a rapid decrease in genetic diversity in Greenland, maybe due to the severe bottleneck effect. And we used admixture analysis to determine the genetic contribution from Canadian Arctic to Greenland. Each individual was estimated with genetic components. With the component, the number of component is denoted by K. So the 4 different color or 7 different color means the proportion of genetic component. So we can say that Greenland, North Greenland population has 5 to 13% of genetic contributions from North Canadian Arctic. So in the future, we hope to reconstruct the muskox lineage, because muskox is the only surviving species of quaternary ovibovine species. There were more muskox-like species, such as Eucerotherium, Surigelia, and Pitherium. Paleontologists, based on their morphology, they grouped into high-horned and low-horned, and Pitherium and Ovibos was classified into low-horned group. That means Pitherium is the closest outgroup of Ovibos, but they are all extinct. And I believe ancient DNA would have some power to reconstruct this kind of muskox lineage. So I want to summarize all my presentation. The first one is which materials could be used for ancient population genomic studies. I would say surface deposited skeletons were a really good source of genetic materials, and horns were the best source of DNA preservation. And the second one, what could be inferred from the whole genome sequence of Greenland muskox, and the Greenland populations were really genetically distinct from Canadian mainland and Arctic, and especially the North Greenland population has some genetic component from North Canadian Arctic. So I would say founder event, bottleneck effect, and all the gene flow collectively shape the population structure in Greenland. So thank you for listening, and special thanks to our sample contributors, collaborators, and our lab members. Thank you.